All right, excellent. Well, thanks, Chris, for the introduction. Sort of the goal for this is to provide a little bit of context about these new products. I know they're a little bit contentious, so I want to sort of start with the first principle science, and then to look more critically at what the opportunities might be. And I'll note that my colleagues at NOPP, I think, will be following up with some specific results from Wisconsin trials in this area. Okay, and just, as always, since we're talking about specific commercial products, just note that we're not making any endorsement of any particular product. This is just, discussion for educational purposes. So we all know, right, our current cropping systems are really, in many cases, reliant on synthetic nitrogen inputs. And this is challenging both from the perspective of farm profitability, right, because, nitrogen fertilizer is expensive, but also, it's fundamentally a relatively inefficient way to deliver nitrogen to our cereal crops, right? If we can get, end use efficiencies of, 75% or more, we're doing pretty good. And that leaves a lot of nitrogen left over that is eventually lost to the environment, either in the form of nitrate leaching, which degrades our water quality, as well as emissions through gases like nitrous oxide that are major drivers of climate change. And so, really a long goal of scientists and agronomists has been able to try to figure out ways that we could do something analogous to what a legume does with respect to nitrogen fixation, right? So legumes are using the rhizobial symbiosis, where we have bacteria in the root nodules of the plant that are able to take nitrogen gas out of the atmosphere and turn it into ammonia, which the plant can use as a major source of nitrogen. As I mentioned, though, with respect to our cereal crops, really, we don't have opportunities for a classical rhizobial symbiosis. And so we're really reliant on application either of synthetic nitrogen or manure that we're obtaining from somewhere else in our cropping system to feed our plants. And so, again, the goal is to see whether we could do something analogous to the rhizobial symbiosis. Could we potentially add additional bacteria to the system, or could we stimulate nitrogen-fixing bacteria that are already present and somehow get them to fix nitrogen in order to be able to back off on our synthetic inputs of nitrogen fertilizer to our systems? And this is really a long-standing goal, right? And I think it's really attractive if we could do this. Potentially we can save money on synthetic nitrogen fertilizer, improve nitrogen use efficiency, and improve some of the environmental impacts that I talked about earlier. So right, it's not necessarily obvious how you might do this. And so we can go out to cropping systems and natural environments, and we can find bacteria that can fix nitrogen, right? There's a lot of diversity out there. However, the challenge is to get these bacteria to actually fix nitrogen in our modern cropping systems. And this is a challenge because the process requires an incredible amount of energy. And so if a bacteria doesn't need to fix nitrogen, it's not going to do it. And I promise this is the only chemical equation I'm going to share today. If you might remember this from an old biology course or something, but to fix nitrogen, right, we take dinitrogen gas, we throw a bunch of ATP into the mix, we have some enzymes as a catalyst, and out pops ammonia. And so you'll notice there's a lot of ATP involved in this reaction. And remember that ATP is the energy currency of the cell, and this is ultimately coming from degradation of sugars or other carbon sources produced by plants. And essentially, right, plants are partly driving these relationships. And if plants are nitrogen starved, they might leak out some more sugars to feed nitrogen fixing bacteria to drive these processes. But if they've got enough nitrogen, they're going to be much stingier with the resources they're going to supply to bacteria to drive these processes. Similarly, right, if bacteria are sensing that lots of nitrogen is available around them in the form of ammonium or nitrate, this is going to downregulate any nitrogen fixation that might be occurring out in the soil or close by to the plant. So we're really sort of stuck if we're trying to drive this process forward in systems that have a lot of nitrogen available, like our modern cereal cropping systems. Final constraint, right? We know that the process of nitrogen fixation is strongly inhibited by oxygen. It turns out that when oxygen interacts with the enzymes that are driving nitrogen fixation, it spoils the process. And for this reason, right, many of you have gone out and seen root nodules of soybean in the field, right? You can crack those open, and you might see something like this image here in the bottom, all of this red pigment, kind of like our own blood. So this is leg hemoglobin. This is a compound that the soybeans are making specifically to scavenge oxygen so that it doesn't impair the process of nitrogen fixation. And so that's why these nodules in soybean are so effective in terms of nitrogen fixation. They're able to tightly regulate the availability of oxygen, right? So cereal crops don't have traditional nodules. And so there are some situations where, plants and microbes can cleverly regulate the availability of oxygen, but this turns out to be another barrier in terms of nitrogen fixation. Okay. Finally, right? Life is tough. It's tough. Life is tough for humans, but maybe even more so for soil microbes because there are all of these other species that you have to compete with. And so when we go out and we survey the microbial diversity of our croplands, there's a tremendous amount of species out there. But what's really interesting is they tend to vary in predictable ways. And so we can measure soil characteristics like pH, carbon, organic matter, texture, and we see that the diversity of microbes that naturally occur, strongly vary with those soil factors. And what this means is that it may really be difficult for newly arriving bacteria to persist and grow in these environments. We might try to add new nitrogen fixers into the equation, but whether or not they'll survive and compete is really another story. And just to quickly share an anecdote, my group led a study where we went out to a sort of classical corn bean field in north central Iowa, and we sampled soils, looked at the bacterial diversity. We did this monthly over 2 years. And what was really striking is that the species that we found in one part of the field tended to persist across that whole study, but they often were really different from species we found just upslope, just 10, 20, 30 feet away. There's a lot of fidelity of these different bacteria to specific soil properties, specific areas of the field. And so you could find evidence, well, maybe a nitrogen fixing bacterium could survive in one part of the field, but maybe not another. And that's really a challenge when we think about altering the microbiology of our cropping systems. And just to drive this home, there was another nice study recently that actually looked at this very question. If you add nitrogen fixers to cropping systems, they found that depending on the field where you looked, there were large differences in the survival of those added nitrogen fixing bacteria. Okay, so how can we do this, right? So we know that there are companies in this space now that are marketing these products. And so what are they actually doing? Well, one sort of general strategy has been to go out, right, to our natural environments, try to select maybe what might be the most effective nitrogen fixing bacteria, bring them back to the lab, isolate them, grow them up, and then sell them back to the agricultural community, right, for application either as a foliar spray or as a seed treatment or as an amendment to a seed. Okay, so I'll talk about a few of these different products that are out there. So one of these is known as Invita. So this involves a bacterium called Gluconoacidobacter diazotrophicus. So this is an endophytic bacteria. So an endophyte is a bacterium that actually lives within the tissue of the plant. It can live inside the leaves or inside the roots. And this can be potentially a good place to live if you're a bacterium, as long as the plant lets you be there. So this particular bacterium was originally isolated in sugarcane, which is, maybe the most ideal environment we might find for having nitrogen fixation within tissues, right? You've got a lot of sugar that a bacteria can use to drive the process of nitrogen fixation. And while they're doing this, right, if they're converting that sugar to ATP, and if they're drawing down the oxygen just simply by burning that sugar quickly, they might create conditions that are actually suitable for nitrogen fixation. So more recently, this bacterium has been found in some other crops, including corn, and so now it's being marketed for use in a range of different cereals. What's one thing that's important to note about many of these bacteria that are currently being marketed is that they oftentimes have multiple traits that may be useful in addition to nitrogen fixation. So this particular bacterium is known to produce some plant hormones, so auxins that can stimulate root growth. It's also been shown to solubilize phosphorus and potentially also to do battle with some fungal pathogens. So in many cases, these bacteria are kind of like Swiss Army knives. They've got sort of diverse traits that might be useful in agriculture. So another product that's out there that you may have heard of is called Utrisha. This uses Methylobacterium symbioticum. This is another endophyte, so a bacteria that lives within a plant cell. This is marketed as a foliar application product. So I'll note that many Methylobacterium, so different members of this sort of broader taxonomic group, are found in plants and soils all over the place. And these kinds of bacteria have also been shown to produce hormones and to solubilize nutrients and to control pathogens. So again, sort of multiple traits that might potentially be useful. Finally, micro-AZ. This is an azospirillum. Two different species of azospirillum are included in this micro-AZ product. And this is an interesting group of bacteria that's been used actually for a couple of decades successfully in South America. It's much more widely used in South America than it is here. So this particular product is marketed as an in-furrow application or a seed treatment. These bacteria are a little bit different from the two that I mentioned above. These are rhizosphere bacteria. So right, the rhizosphere is the zone right around the roots. So these are bacteria that are typically living in close association with the plant root. So these particular organisms have been shown pretty convincingly to stimulate root growth, which might be one of the reasons that they benefit crop production in addition to supplying some nitrogen. And what's interesting, right? If you look at a whole synthesis of different plot and field scale studies throughout Brazil, there was pretty clear evidence of a 3 to 5% average corn yield benefit among years. So some fairly convincing evidence that these bacteria can benefit corn production in Brazil. Jury is still out as to whether they might be effective here in the upper Midwest. I'm not aware of any peer-reviewed studies that have convincingly shown them to increase yield in our kinds of environments. Okay, so another sort of class of these different nitrogen fixing products involves taking bacteria and actually gene editing them to alter their functions. And so one example here that many of you might have heard of is the Pivot Bio microbes. And so they have a gene editing technology where they're actually able to remove some of the constraints on nitrogen fixation. So I mentioned earlier that if bacteria sense mineral nitrogen in their surrounding environment, they typically downregulate nitrogen fixation. Well, in this case, they've been able to take the brakes off, and that allows these bacteria to keep fixing nitrogen even when there's a surplus of nitrogen in their environment. In some cases, they've also tinkered with the genetics to allow those bacteria to leak nitrogen out of their cells, right? That's another thing that ordinarily a bacterium doesn't want to do, right? It wants to keep the nitrogen for itself. I'll note, though, that these aren't transgenic organisms because they're not, adding genes from other organisms. They're tinkering with the genetics that was already present to begin with. So this is marketed as an in-furrow application or a seed treatment. I'll also note that, some people see the name Klebsiella and immediately, right, you think mastitis and other kinds of, pathogens. Note they've done some testing on this particular Klebsiella species and shown that it's not a pathogen. That's one of the interesting facts of microbiology, right? You can have closely related bacteria that have really different kind of ecological functions in the environment. Also note, just as the bacteria I mentioned a few minutes ago, these particular ones are also known to produce plant hormones that can promote root growth. So some of the effects that we see might be due to changes in root morphology. So there's some pretty convincing scientific evidence that this gene engineering can actually can indeed increase nitrogen fixation. And so here's an example where they show that this gene-edited Klebsiella can release a fair amount of ammonium to solution in the lab. This figure shows these multiple strains of Klebsiella, and the gene-edited strain is indeed able to release them. Okay, so maybe it works in the lab, maybe in the greenhouse, right? The challenge is, well, can this actually work under field conditions? And I think that's really where the controversy and the scientific uncertainty begins to set in. And so there was one study, I'll note the caveat, this is an industry funded study, so right, take it with a grain of salt, but a number of different large plot trials across the Midwest over 2018 and 2019, variable response as you might predict. They found about 70% of the plots had a yield increase with these gene edited microbes. So they found on average about a 3% yield increase. So I think this is plausible. This is same order of magnitude of what studies have suggested from some of these products in South America. So I think it is plausible. One thing I'll note is that you'll probably remember 2018 and 2019 were pretty wet years across much of the Corn Belt. 2018 in particular, right, was a really wet year. And that, sort of begs the question, well, how important are these environmental factors actually in driving performance of these microbes? And so bring this back. And this is important because many of you are aware that sort of subsequent work throughout the Corn Belt done by extension scientists has shown really very little, if any, yield benefit from adding some of these biological processes. However, right, a lot of this work was happening during really dry years throughout the region. So we would expect, just from first principles, that differences in soil moisture might have a large impact on biological nitrogen fixation, right? So we all know when you wet a soil up, it's much more challenging for oxygen to diffuse from the atmosphere into that soil. And as a consequence, right, we can have these little zones in the soil, particularly close to roots, right, where there's a lot of microbial activity consuming oxygen, where we can have these oxygen depleted or anaerobic sites where potentially, right, nitrogen fixers might be really happy, right? We don't have this inhibitory effect of oxygen on nitrogen fixation. Whereas under drier conditions, right, it's much easier for gases to diffuse through soil. It's much more challenging if you're a bacteria trying to fix nitrogen. You've got oxygen all around you that's potentially poisoning the process. So I think that's one really interesting question, the degree to which, moisture is really a constraint on this relationship. I'm excited that a couple of my colleagues in Extension, Dan Marzu and Chris Bandura, are doing some trials this year, or just ideally just finishing these trials up at Marshfield, where they tested these products under the wet conditions that we had this year. So really curious if we might finally be able to see some response of these biological products under these wet soil conditions. Okay, so another strategy that I think is really interesting and that is likely to emerge, over the next few years is looking at nitrogen fixation from the side of the plants, right? Can we gene edit or breed for plant traits that can actually promote nitrogen fixation bacteria? So one really exciting observation made a few years ago is that there are some corn plants in southern Mexico that have aerial roots that naturally produce a mucilage that is a great habitat for nitrogen-fixing organisms. This mucilage is really rich in sugars, right? So it's great food for nitrogen fixers. And it also sets up a barrier for oxygen where the nitrogen fixation can happen in a lower oxygen environment. So one of the real leaders of this research area is Jean-Michel Anet at UW, who's actively working on this question. And optimistically, we might have hybrids with these kinds of traits that would be suited for Midwestern conditions within the next few years. So stay tuned. Not quite ready for prime time, but really promising to actually couple plant traits with these nitrogen fixing organisms. Okay, so as I mentioned, there's a lot of uncertainty. A lot of these products are being marketed, but a lot of the fundamental scientific questions are still not necessarily clear, right? How much nitrogen is actually fixed, if anything? To get at this, we need really robust nitrogen rate trials, which some of my colleagues with NOPP and some collaborators around the state have been leading. Unfortunately, not great news yet in that regard. In cases where yield does change, right, I think we also need to think about what the mode of action is. For some of these other bacteria, it seems actually that changes in root traits and other factors might even be more important than nitrogen fixation. And finally, how does plant genetics, environment, and management really interact to shape the response. And I think there's, really exciting opportunity for leveraging our understanding of soil ecology really to drive these processes forward in the future, right? You might expect, well, we might have much more custom mixes of microbes that might be tailored for certain soil types or environmental conditions. It may be that we have to think about feeding the microbes that we add, right, so that they can survive before they can start colonizing the root. Maybe that there are, specific combinations of hybrids and microbes that we can use to best drive this process forward. And it might be that certain conservation practices might be well suited for these kinds of amendments, right? If we reduce tillage, we know that we can actually generate these soil aggregates where there's less oxygen diffusion. These could be great places for nitrogen fixation to happen. So take-home points. The jury's still out. There's some evidence, scattered evidence that we can actually increase nitrogen fixation in cereal crops with some of these microbial amendments, but it's not by any means consistent. The existing products really differ in important ways. It's not clear which approach may be best yet. And we still don't know why some of these products succeed or fail in certain fields in certain years. So, I urge people to keep an open mind. If you tried a product and it didn't work, don't necessarily write off the whole field. I think there's going to be a lot of movement in this space over the coming years. So, yeah, that's all I've got. Happy to take questions if there are any.
Yeah, we had one participant raise their hand. I'm not sure if that was accidental or not, but if you want to unmute and ask your question. We did put into the chat another document that supports some of the information that Stephen talked about. It's kind of a review of the different nitrogen biological products from UW Extension's Nutrient and Pest Management team. So you can find that in the chat. Any questions? Yeah, is it really cool, direction that agronomy is taking? When I went to college and learned crops and soils, it was feed the crop. And now it's really cool to see things shifting toward if we feed the soil, it will feed the crop as well. So it's cool, it's exciting, but also know that, a lot of this research is in its infancy and is very complex. It's not just like throwing fertilizer on there and getting a yield response. Very, very unique processes and tied to the environment too. Thank you, Stephen.